All right, here are some enrichment problems on DNA. Now, we didn't actually spend class time doing these here on campus, but I gave them to the students on campus as just something to do on your own time if you want the extra enrichment on DNA. A lot of students don't tend to struggle too much with DNA because you've done it before in high school and in Biology 1. But if it's been a while since you took Bio 1 or high school biology, I do make the DNA enrichment and I give it to students just as something optional, but we don't spend time on campus doing it. It's just something optional, something extra. So these are the problems that we did. You can download this file from the course website, or you can just pause the recording and then try these for yourself and then hit resume when you're ready to go over it. Here are problems 1 through 7. Pause, try it for yourself, and then go over it. Try not to use your notes. Okay, that's what I recommend. I recommend trying to do this completely from memory, because if you do it from your notes, well, then you're really not testing your ability to recall the information. You're just testing your ability to read your notes. So I recommend trying to do this without your notes first, and then if you get stuck, only use your notes as a last resort. There's 1 through 7, and then there are 8 and 9. So again, this was something I gave to my students on campus. Here's just something bonus. And so I'm going to go over them now so that everybody's got the answers. And the written PDF answer key is also posted on the course website. So let's go through these. Compare and contrast DNA versus RNA. All right, so DNA and RNA, oops, didn't mean to do that. DNA and RNA do have several things in common, but they are more different than they are similar. So what do they have in common? Well, the similarities, they are both polymers of nucleotides. Right, nucleotides, not nucleosides, linked by phosphodiester bonds. And that's pretty much where they stop being common after that. So let's talk about what they have different, because there are more differences than there are similarities. Right, DNA is single-stranded. Whereas RNA is double-stranded. I wrote that backwards. DNA is double-stranded. Whereas RNA is single-stranded. Right? RNA is composed of 1 strand, whereas DNA is composed of 2 strands. Let's see, DNA contains deoxyribose as its sugar, whereas RNA just contains ribose. And this is kind of getting into something, I think I put it in number 2. Yes, number 2 talks about the sugar. So we won't draw it out right now because that's the question that we're going to do next. And then in terms of the bases are A, T, G, and C, whereas the bases in RNA are A, U, G, and C. Okay, so they both contain A, both contain G, both contain C, but one contains thymine, one contains uracil, and the only difference here is that this one's got a methyl group that this one doesn't have. So it's not like they're hugely different in terms of the uracil part, but one contains thymine, whereas one contains uracil. But in terms of their similarities, they're both polymers of nucleotides linked by phosphodiester linkages. All right, so that was number 1. Let's look at number 2. All right, draw the pentose sugar found in RNA, then draw the pentose sugar found in DNA, and number them, label which is which, and how do they differ. All right, so the sugar, it is a pentose, specifically it's ribose, versus deoxyribose, okay? That's the difference. One's ribose, one's deoxyribose. And so deoxyribose is a 5-membered ring. And remember, we use the primes to delineate from the carbons in the base. So 1 prime, 2 prime, 3 prime, 4 prime, and there's 5 prime. Make my O a little bit smaller there. Okay. And so they're both 5-membered rings. They both look that same general structure. 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. Okay, they're both in the beta configuration. So they're both like this. Let's just distinguish this one as being DNA. This one is RNA. So this is just ribose, regular old ribose, whereas this is deoxyribose. And what makes it deoxy is that carbon number 2 has no OH group. Okay, it's just 2 hydrogens. Right? In DNA, it's got deoxyribose. That means there's no OH group on the second carbon. Whereas in RNA, it does have that OH group. Okay, so that's the big difference. Deoxyribose versus ribose. And then the rest of it's the same. Okay, deoxyribose versus ribose. Deoxyribose is the sugar of DNA versus ribose is the sugar of RNA. Hence the name deoxyribonucleic acid versus ribonucleic acid. And they're both pentose. Pentose just means 5. Right. All right. Let's go on to number 3. In the primary structure of DNA, where does the bond form between which 2 atoms to make the DNA polymer? All right, so between which 2 atoms? This is occurring between the 3' OH group and the phosphate on the 5' carbon. Okay, it's between the 3' OH group and the 5' carbon. So if you've got, I'm going to try to draw this as best I can. Right, because that's phosphate. There's 5'. Okay, this is 1', 2', 3', 4'. Here's where your base would be. So A, T, G, or C. Or uracil if we're talking about RNA. Right, because uracil is going to have this, RNA is going to have the same structure. So it's between this OH group and then the phosphate beneath it. So PO3. Oop, that wouldn't be 3 minus, that would be 2 minus. 2 minus O. And then there's the rest of the chain again. One prime, 2 prime, 3 prime, 4 prime, 5 prime. So it's between this phosphate and this OH. So if you actually draw the phosphate out, think back to your Gen Chem days, that's where you're getting the bond. Okay, so the phosphodiester bond is occurring right here. This whole thing collectively will be the phosphodiester bond. So when we draw it, there's my 3. You've got O, and then PO2 minus O CH2O. And here's your base, whatever it is. And then there's 2 prime, 3 prime. And so this collectively is your phosphodiester bond. And so you've got a free 5 prime end, and then you've got a free 3 prime end. So no matter how many bases you have, whether it's just one phosphodiester bond or 6,000, right, you're always going to have one free 5 prime end, you're always going to have one free 3 prime end. So the phosphodiester bond is occurring between the 3 prime OH group and the phosphate of the 5 prime carbon. Right, so that's where your phosphodiester bond is occurring. So now this one's free to bond to another phosphate group. This one's free to bond to another OH group. So you've always got room for growth on both ends. So that's what's happening to make the phosphodiester bond. All right, number 4, how do you sequence DNA? What are you doing? All right, so what you're doing here is you are reading the bases only, not the sugar, phosphate, backbone, right? Because that's in common. So if you've got your backbone, right, we're just gonna sketch it kind of like what your book does, because it does kind of look zigzaggy, right? Let's just go T, G, C, A, C, A, right? What you're doing is you're listing the bases. So this will be sequenced, oops, T, G, C, A, C, A. And then at which end do you begin? You always begin at the 5 prime end, and you read 5 prime to 3 prime. Always read 5 prime to 3 prime. So you begin at 5 prime end, and which direction do you go? You read 5 prime to 3 prime, always. So you're not actually sequencing the backbone, right? You're just sequencing what those bases are. Starting at the 5 prime, ending at the 3 prime. That's the convention. No matter where you are in the world, you're going to read 5' to 3'. All right. Number 5, I don't think I need to skip a whole page for that because that's pretty easy to answer. Don't need a whole lot of room. 2 strands of DNA are held together by what? Held together by hydrogen bonds. And that's significant because hydrogen bonds are an IMF, intermolecular force, not a covalent bond. Right? A lot of textbooks show DNA as like these ladder rungs, like it's just rock solid. But it's actually held together by intermolecular forces. A and T, that's 2 hydrogen bonds. And G and C have 3 hydrogen bonds. Okay, so collectively those hydrogen bonds are strong, right? If you've got a sequence of 10 million, right? Collectively those hydrogen bonds do all add up, but I want you to know that there's no sharing of electrons going on here, right? These are not covalent bonds. These are intermolecular forces, and that's what's significant. All right, I think I've got enough room to answer this question as well without making a whole bunch of space. I'll just make that font a little bit bigger. There we go. So if this is the DNA, 5' to 3', then that means that the complementary strand would be 3' to 5', right? Because they run anti-parallel. It's like one's going east and one's going west, right? They run anti-parallel. And then all you would need to do is list what's on the other side. So the complement of A is T, complement of G is C. So then T, then A, then G, then G, then G, then T, then A, then T, then C. Started having to squish my letters there, right? A and T, G and C, A, T, A, T, G, C, G, C, G, C, A, T, T, A, T, G, C. Right, so if you just remember, A is complementary to T, and G is complementary to C, and then remember that the other one is anti-parallel, right? So that means run in opposite directions. So that's what that means. That would be the complementary strand. And again, I know a lot of this is stuff that you did in high school biology, but it's always good to review, and it's always good to make sure that you're caught up to speed to the level I want you to know it for this class. All right. Let's give myself some room to write here. There we go. State the role of each of these in DNA replication. So, helicase, what is its job? Its job is to unwind the DNA at the replication fork. And there can be multiple replication forks going on at one time, right? If there's the DNA, you have a helicase sitting here unwinding it. Right? There's the replication fork, and then there would be the helicase. All right, polymerase, it synthesizes new phosphodiester bonds in newly forming strands. Right, so there would be a polymerase anywhere you're forming new bonds, right? The polymerase is going to be moving down the line. And then there's one maybe leading, one that's lagging. Right? The polymerase is the protein that's making those new phosphodiester bonds. All right, ligase is a protein that seals phosphodiester bonds in the Okazaki fragments of lagging strand. Plus, it also seals after topoisomerase breaks. Okay, so there's going to have to be a ligase sitting here to seal all these back up. And so what does topoisomerase do? It makes single or double, depending if it's topoisomerase 1 or 2, stranded breaks in the DNA as it's unwinding to remove any sort of knots, tangles, etc. Right? Because if you've got a twist, you can't have this process taking place. And then primase, this adds an RNA primer. So you have to have a primase here as well to begin synthesis of new strands. So you'd have to have a primase for every one of these Okazaki fragments as well, and then all those primers have to be removed, and then it's all put back together, right? So it's a messy process. It's a big, nasty, messy process, and there's a lot involved, okay? And a lot of textbooks kind of, in like high school, oversimplify it and just say, oh, it splits open, this stuff goes in, then it closes back up, and it's done, right? But in high school, you just look at the big picture. And even here, this is just elementary of biochem. When you take full semester biochem, you'll get into even more detail than you probably ever wanted to know about all this stuff too. So we're just kind of getting our feet slowly into the water here. And then the deeper you go in your studies, the more detail you will learn about your molecular biology. So lots of vocab. All right. Explain the difference between the leading and the lagging strands. And you can draw a picture if that helps. And I think it does. I think it definitely helps to draw a picture. Although I don't claim to be good at art, I think a picture is definitely worth 1,000 words. So replication only occurs 5' to 3'. So if you've got your parent DNA, all right, let's make ourselves a big replication fork just to make it plenty of room to draw. Okay, so let's just arbitrarily choose this one to be 5', this one to be 3'. All right, so leading strand is synthesized continuously whereas lagging strand is synthesized discontinuously. All right, so what does that mean? Remember when we talked about the process of DNA replication occurring only in one direction at a time, right? That means that we can only go 5' to 3' at once. We can't go, one side's going 5' to 3', the other side's going 3' to 5'. That doesn't work. So if 3''s here on the parent strand, and 5''s here on the parent strand, that means that the new strand, 5''s going to be up here, right? So you can have your polymerase attach and just go down the line. Nice and smooth, right? This would be the leading strand. Because polymerase can just attach here and just go down the line, right? Because if this is 3', that means that 5''s up here on the opposite strand. So it's just going down the line, nice and smooth. All right. But the other one, to have 3', 3' here, 5' up here. You know, I wrote that backwards. Drew it starting at the wrong spot. Here's 5'. There's where your primase, and there's where your polymerase will be, and it'll just be going. There you go. Now your other one is going to be your discontinuous one. So you're going to have a polymerase start here and go, and then, oops. I'm not a very good artist, can you tell? If 5''s up here, you'll have a polymerase start here and go, and then one here and go, and then one here and go. Right? It's a lot messier. So this is your lagging strand. And the figures in your textbook are a lot better at drawing this than I am. One's synthesized continuously, and one's synthesized discontinuously, because you can only go in one direction at a time. And I'm sorry for my struggles in drawing that. I'm not very artistically inclined. So my advice to you is to consult your textbook for a better picture. And then the last one, let's do some mutations here. Let's give myself some room. All right. So if this is the parent strand, let's see what would happen if all these things occurred. So let's just number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay, if an insertion mutation occurred after the 4th base. So an insertion mutation occurs when you have extra base or bases added. Right? So if we've got an insertion mutation, we're going to draw the same thing we had originally after the 4th base. So AGAT, AGAT, right? There's 1, 2, 3, 4. And then there's an insertion mutation. So I didn't tell you what to insert, so you can just make up whatever you want. Let's just go with an extra T. T got duplicated, right? That would be your insertion mutation. And then there's the rest of the original sequence. So C, 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 A, T, A, G. 5, 6, 7, 8, 9, 10, 11. Right? It was originally 11. Now it's 12 because it's got that extra base that's been stuck in there. All right, a point mutation occurs when you've got a base that is incorrectly substituted. I guess I need to be able to see the parent DNA, huh? Let's just leave it like it was. Point mutation occurs on the 7th base. So a point mutation is where you've got a wrong substitution. Okay, so let's draw it out what our genome should be. A, G, A, T, C, 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 A, T, A, G. And it's on the 7th base. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So a point mutation is where you've got a wrong substitution. So let's just pretend that that C now becomes a T. Number 7. That would be your point mutation right there. Wrong substitution. And now for the last one, I'm going to recopy the parent DNA. What it should be, A, G, A, T, C, 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 A, T, A, G. So that when I copy it up, I can see it. A deletion mutation occurred on the 5th base. Okay. So a deletion mutation means you lose a base or a base is. Okay. So if we've got A, G, A, T, C, 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 A, T, A, G. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. And we've got a deletion. That means that number 5 got deleted. Right? So what does that give us? A, G, A, T, C, C, A, T, A, G. Right? It goes 1, 2, 3, 4, 6, 7, 8, 9, 10, 11. You've lost the 5th base. All right, so that's what deletion mutation looks like. So those are the enrichment problems. Again, I know most of this is review of your high school and Bio 1, but just to make sure that we're all caught up where we need to be, that's the end of this enrichment.